Thank you very much for all coming today. I'd like to welcome you to our workshop on innovative phase 3 randomized trials that address multiple research questions. My name is Max Parmar. I'm the director of the MRC Clinical Trials Unit at UCL and the Institute of Clinical Trials and Methodology. And I'm supposed to tell you also that for those who are more technologically minded, that we've got a hashtag for a tweet there. So if you want to tweet today, you can, and using that hashtag. So the motivation for today was really to have a day where we discussed some of the new designs that are being developed in this area, particularly those that are coming out of the MRC Clinical Trials Unit, and discuss some of the rationale for them and give you some examples and have a discussion about both their application and how they can be, what other designs we might need in these sorts of areas. And what I thought I would do would be just to start to give you a little bit of background and a little bit of context of why we think we need new designs in clinical trials. And a lot of you, I'm looking around, and I know a number of you in the audience will be familiar with some of this. But just to start from the top here, we know that the development and testing process for interventions is just too slow. If you talk to people, that's 10 years, up to 20 years before a new intervention is brought into routine practice. And even worse than that, what we find is when we find a new therapy is not better, which is really quite often, somewhere between 50, 60, 70% of the time, depending on your disease area. Too often we're showing that right at the end of the process in the phase 3 trial. And so we've spent years to get there, and then we find we actually have made no progress. And as a consequence of that, there are many diseases we've found that we've made little or very slow progress over many years, over decades often. What's changed now is that actually, you know, again, looking around, there are one or two who are a bit longer in the tooth than others, and a bit like me, have been around, and know that the numbers of new interventions now demanding evaluation is much larger than it was 15 years ago. So the actual problem has changed for us. And we also know that a lot of trialists in the audience, we also know that the process of developing and starting a new trial is very time consuming, and there's often a long gap between trials. And so we need a step change in efficiency. We don't need to just tinker around the edges. We need a step change in what we're doing in efficiency. And a key part of this step change has been platform trials, master protocols. The language here is a little bit loose, and it hasn't, you know, you talk to different people and the definitions of these things are different, but I don't think it matters that much. And what we're going to talk about today are four specific designs that we've developed at the MRC-CT: the MAMS platform design, the MAMS selection design, the MAMS-ROCI design, and the practical design. And the common element to all of them is that many primary hypotheses are in one protocol. That's the common element. And often some sort of adaptation, dropping arms, adding further primary hypotheses. So that's quite a useful way to think about these things. And many of these solutions, if you look at the literature, a lot of the solutions that you see for this sort of problem of addressing many questions in the primary hypothesis, trying to do a step change in efficiency, has been done in the phase 2 context. The literature is full of improvements in phase 2 trials, trying to do these sorts of things. Our own view is this will have limited impact. They're good, but they have limited impact, and I'll try to explain why that's the case. And the reason for that is really quite simple. The problem has been historically, not that we haven't had signals at phase 2, that we've had actually quite a lot of phase 2 trials which have shown a positive signals. What's happened is those phase 2 signals, the improvements at the phase 2, have not translated into improvements, changes in phase 3 outcomes. So the improvements we're making in design and efficiency at phase 2 will just lead to more signals. The failure rate will remain the same because we haven't changed the signal. So the obvious answer is change the signal, of course, isn't it? Of course, but that's not going to solve the problem because you don't know that that signal is any better than the old signal that you have. And the only way of assessing that is by doing trials which relate the treatment effect on that signal to the treatment effect on the phase 3 outcome, where both are positive. Well, once you've done all of that, that will be many, many, many decades, if not generations. So we've got ourselves into a real bind, I think. We have to deal with that problem. So the step change that we need can only be achieved by amending the phase 3 part of the process, not the phase 2 part of the process. It's the longest, most expensive part of the evaluation process. And an example, I'll just give you an example of this in Parkinson's disease. I'm very grateful to Tom Faultley and Camille Carroll. Camille is in the audience today, who've been working on this Parkinson's disease, and for these 2 slides. A lot of you will know people with Parkinson's disease, and we know that in that area, there have been lots of trials, so that the brown blobs, or the sort of reddish-brown blobs are the phase 2 trials. Lots of phase 2 trials showing signals. Subsequently, the yellow blocks are the phase 3 trials. And the phase 3 trials do not show an improvement in outcomes. So it's not that we haven't had the signals, they just haven't translated. And this is the consistent message that we get. And of course, we know that with Parkinson's disease at the moment, we only have symptomatic, we don't have disease modification treatments. And we in academia, most of the people here from academia, I can see 1 or 2, I think, who are industry partners, have a unique opportunity to contribute to this area, and try and describe why. Firstly, all of the designs that we're going to describe to you test many of interventions. They're either newish interventions, repurposed interventions, the same treatment but at different doses or durations, many different doses or durations, or they're looking at many standard treatments. But the common element is they've got many things all at the same time being changed. And if you think about it, industry will only be ever interested, usually, at any given time in a single treatment for a given disease. It's not in their interest to have many treatments for the same disease. Commercially, it doesn't make sense, of course. So academia is uniquely placed to do these trials where you have many interventions, or many interventions at different doses or durations, because industry is not going to play in that space. And experience supports this, because most of the trials doing assessing many interventions at the same time are done by academia, not by industry. Industry collaborates in them. There are some done by industry, but that's pretty rare. So I'll just finish this section to give you a little bit of the feel for the day coming towards you. Is the 4 new randomized phase 3 designs we're talking about will go in terms of order of maturity. The first one we'll talk about is the multi-arm, multi-stage platform design. That design goes back to about 2000, so it's over 20 years ago. And the key characteristics of that design is that it's a more efficient superiority design, i.e. looking for superiority. Usually, there's a single standard of care or control arm. You're testing many interventions. At various stages, you're dropping arms or adding arms. And all arms, in theory, can recruit enough patients to actually be analyzed to the primary outcome measure. So they're the key characteristics. Just hang on to those key characteristics of that design. The next design that we'll be talking about in the second session, or in the second part of the MAMS session, will be the multi-arm, multi-stage selection designs. Same as the MAMS trial, the multi-arm, multi-stage trial, but you limit the number of arms going through that at each stage, because you've either got a limited number of patients, you've got more limited funding, or there are timeline constraints. Not all of the arms can possibly go through the end. It's just too big, too expensive, or too many patients. The third design that we'll be talking about today is the multi-stage response over continuous outcome design. It's a bit of a mouthful, but this is just one way of doing a more efficient non-inferiority trial. So what you're doing, rather than testing one non-inferiority intervention, or one way of reducing a dose, or one way of reducing a duration, just test many of them. How can you do that efficiently? You're testing many durations or doses of an intervention or a drug. And how can you do that efficiently? And you'll hear about that. Again, in this case, you've got a single standard of care or control arm, which is the standard dose or standard duration. And the final one that we'll be talking about is the practical design, the personalized randomized controlled design. This is quite a different situation when there are a large number of possible treatments available for an individual. They're all relatively standard treatments. A good example of that is the antibiotic situation, where you might have a number of antibiotics available for the treatment of a certain condition. But the key here is that each individual, as they come in, is only eligible for a proportion of the eligible treatments, depending on their own characteristics and the site choices or the site availability of those treatments, depending on the country or the individual hospital. In this case, there's no clear standard of care arm, no control arm, and the aim is to get a ranking of the many treatments that you're considering and say, how do we choose between all of these treatments? And again, you'll hear about this today. The aim of this workshop is to present key aspects of these designs and key components of these designs and their thinking and some of the statistical issues they raise. Particularly important, what we're going to do is present some real examples. There's plenty of time for discussion. At the end of each session, we've got about nearly, I think, 25 minutes for discussion on these things. So plenty of time discussions, questions and comments. There's the event program, and recordings will be available later on the same page. So we've got a recording going on, and that will be available for people. For those who don't want to ask questions through the microphone, you can log on to the Mentimeter at the start of each panel discussion, and you can submit questions via that, and they will be asked as well. So you can do it both ways, or you can ask in the traditional way. And I will stop there. I'm happy to answer any questions of fact or information at this stage. Any questions of interpretation or general discussion, we can help pick up in the panel discussion. No, should we get started? I'll pass on to Jeremy Chataway. Jeremy, you might introduce yourself, and then we can start with the first session. Here, you can have this. I'll go to that.
Great, thanks very much. So I'm not a statistician, so you have to be kind to me. So with session one, you can see in front of you, and we had 4 very nice talks that we'll go through. And as I said, any facts after each talk, then room for 1 or 2 points, but then we'll save it all for the 25-minute panel discussion at the end. So it really saves up all your questions and questions that come in online. And then after that, we'll break for lunch, and then we'll carry on. So, of course, it gives me great pleasure to introduce Max. So Max is going to give the first presentation on MAMS platform designs. Thank you, Max.
So I've already given you a little bit of motivation for this. I won't go through the motivation again for this sort of design. I hope I've persuaded you to just have a step check of what we're doing. Otherwise, you won't make progress in our lifetime. Can you hear me? Better? No. Hold on. Yeah, that's better. It just needs to be a bit closer. Okay. So, in this design, the first thing to emphasize is what we're doing is testing many interventions. So on the left-hand side, you've got what we traditionally do, the 2-arm trials of control against each research arm. Very simply, if you put them all together, you can have control versus research A, B, C, and D, and you can see immediately you get a benefit in that because you've only got one control arm and you're evaluating all things at once. It doesn't take a a genius to do that. Then you can add a multi-stage element to that. At each day, at various stages along the way, you can compare T1 versus C, T2 versus C, T3 versus C, and T4 versus C. And think of these as pairwise comparisons. So one way of thinking of this design is just very efficient way of doing a lot of two-arm trials. And at each interim stage, you ask the question, shall we continue testing this or investigating this new intervention? Shall we keep testing this intervention? And you can think of the early stages like a phase 2 trial. Isn't quite like that, but you can think of them like that. And I don't know why I show this, because it's obvious, isn't it? It's just faster. The cost is going to be less, and it's more flexible. You can drop and add arms. I don't know why we need to work out how much faster. It's just obvious, isn't it? So let's give you an example of this, of STAMPEDE. A lot of you will be aware of this trial in prostate cancer. When we started this, there were about 1 million new prostate cancers a year, about 2000. Now there are about 1.3 million new prostate cancers worldwide. When we started, there had been no progress in the treatment of men with metastatic or for high-risk prostate cancer, I should be clear, locally advanced disease or metastatic disease, for 40 years. Hormone therapy had been the standard of care, and it hadn't changed, which is just, I don't know how we got there. Well, I know how we got there. We'd actually done 2-arm trials which had not made any progress. And median survival for men with metastatic disease was about 3 years, with high-risk locally advanced disease a bit longer, about 5-6 years. And we thought, well, can we change the game here? So we launched STAMPEDE in 2005. We launched STAMPEDE in October 2005 was when we actually started recruiting patients to this trial. And we started recruiting to 6 arms, 1 control arm. So at the top here, you've got the standard of care arm, which goes through from 2005 all the way up to 2029. So it's a long trial, isn't it? And we had 6 arms, 5 research arms, B through to F. I won't go into the details of those arms today. And the solid blocks told you the period of accrual. The dotted lines show you the follow-up. And then the solid block at the end of each line shows you the time point of the primary analysis, of comparing B versus A, C versus A, D versus A, and so on and so forth. Over time, starting in 2011, we've added 5 further arms. From 2011 to 2017, we've added 5 further arms. So that's 10 research arms added from 2005 to 2017, so a period of 12 years. And we're going to add further arms. 3, that looks like lots of arms, but it's actually 3 further arms done in a factorial design, some of it. And that they will be added into STAMPEDE 2 rather than STAMPEDE 1. And we can discuss why that's the case. And this shows you the accrual. Each dot here is a patient. As you can see, it started relatively slowly, and then it's picked up over time. And you can see when we've made various changes and additions. And you can see down at the bottom, at each time, we've changed standard of care. And you can see there that we've changed standard care 5 times, once due to results external to STAMPEDE, and 4 from the consequence of results of STAMPEDE itself, and other trials which are asking the same question. So at each stage, at each of those times, we've changed the standard of care within STAMPEDE, added that standard of care both to the control arm and to the research arms, and routine care internationally has changed as well. So this is a summary of it, that we've had 12,000 participants from more than 120 sites, 4 changes in standard of care as a consequence of Stampede itself, and for men with metastatic disease, median survival has gone from about 3 years to about 7 years. Now, if you think of any other way that we were going to try and do that over a 20-year period, we weren't going to achieve that, of course. Interestingly, we've had those 10 interventions evaluated in those 20 years. Chemotherapy, radiotherapy, hormone therapy, a COX-2 inhibitor, a bisphosphonate, and metformin, all different types of agents or interventions, a mixture of repurposed and commercial drugs under patent. And because of that investment over time, a new result emerges every 12 to 18 months because the arms are maturing approximately every year or year and a half. So there's still results to come yet from the 2 arms which haven't reported. One of the reasons you do these sorts of trials is not just the statistical benefits, but the operational benefits. And this plot shows you when we first started Stampede, with the first 6 arms, how long it took us. So that plot shows you 60 months, which is 5 years, for 120 centers to get their first patient in. And we all know who do trials, and it takes forever, doesn't it, to get these trials off the ground. And for centers, and things, centers say, oh yeah, we're coming on. And 2 years later, they're on board. And even worse now, of course, because we're in such a terrible state in terms of research, infrastructure. So it's takes it forever. But for all of the new arms, within 6 months, they're up and running with their first patient. So you can see the operational benefits here very, very clearly. For the rest of the talk, what I'll do is talk a little bit about some of the practical strategic issues and some of the design statistical issues, although Babak will concentrate more on the statistical issues in his presentation. Because these are challenging to develop, fund and run. More than 4,000 individuals have been involved over those 20-year periods, 9 funders from government, charities and industry. And of course, there are voices out there which say, actually, that you're just cornering the market in research here. It is the trial, while you're just excluding others doing things in this area. Well, yes, that's probably true, but the prize is so big here, the opportunity to make progress is so large that it's worth that conflict. And what's key to the discussion here, and we'll hear a little bit from a patient partner later on in the session, is the patients were key in getting this through, in the development, design, funding. And when we had the first discussions in about 2000 at a committee, I won't tell you the funding committee, the reason it got through that funding committee was the patients said, why aren't we doing this? Because of course at that stage it just looked mad. The good thing is the development process and the running of these things needs a wide range of expertise, so you can draw many people in, but not everybody is going to buy into this. You don't need everybody, actually. You just got to get enough. Hundreds and hundreds of people involved. And as I say, not everyone is bought into these trials. You just need enough to get started. If you try and get everybody on board, you'll never get started. And people will continue to throw rocks at these things. And you can understand that from one perspective, of course. But as a research community, I think we need to assess what we can do as a group rather than as individuals. As I've shown you with Stampede, funding often needs a number of sources. And of course, if you aim to the key is to try and get off the ground. What do you need to get started? And actually, all you need to get started is 2 research arms. And often, and I'll come back to why it's good to get started with repurposed arms in a minute. Get started with at least 2 research arms. And then all you need is a line in your protocol saying, we will add further research arms as the trial continues. That's all you need to say. If you put that in your protocol, any funders here, you don't even need to tell the funders you're going to do that because they'll never read the protocol. You send that to the regulators, and because you put it in your protocol, you can do it. You don't need to say how you're going to do it, when you're going to do it, just add it. And they will review it, and once they've reviewed it, it's been approved. Of course, they won't read every word. What about industry? People will say, what about industry? Actually, our experience is, it's not always the case, but actually a good place to start is with repurposed arms. The challenge with interacting with industry right at the early stages is they've got very, very deep pockets. And actually, they can drive the agenda. And what you want to do is to build a structure that will encourage them to come forward, but you're in a strong position for the negotiation. You're not in a strong position when you're getting started, because it's really quite difficult to get any trial started. We know that. What we've found, if you build that infrastructure, they will come. One of the things you shouldn't underestimate is how hard this is on the central coordinating team and the site trial teams. This is really, really hard to do because you're doing lots of things at once. And one way of thinking about that is a two-arm trial is like just juggling two balls. Here, what you're doing is picking up any a sort of 8, 10 balls all at the same time and juggling those. Everybody is doing that, both at the central coordinating center and at the site trial, at the sites, because they're all doing closing arms, sending SAEs on different arms. Forms, of course, can become a bit of a mess, so you can see how this gets really quite complicated for everybody involved. And there is a real benefit of doing repurposed drugs. Repurposed drugs have got a bad sort of rap often in the scientific community. Actually, industry repurposes all the time. Industry repurposes loads and loads of drugs. Monoclonal antibodies are a very good example. Started off in oncology. They now use them in multiple sclerosis, in arthritis, a range of other areas. They just repurpose their drugs, call them as something else, and sometimes even charge more for them. So there's loads of examples where repurposed drugs have been used in a wide variety. I don't need to go through all of these examples for you. You know, COVID-19 is a very good example. Most of the drugs that are effective, well, a lot of the new drugs don't work actually today. It's all the old drugs that work. And in multiple sclerosis, most of the drugs that do work in relapsing remitting disease are repurposed agents. One of the practical things we've done is have a new comparison chief investigator for each new arm. That can help both politically, because it allows you to bring more people into the fold, but also it helps work-wise as well. And if you can do that in a transparent and open way, even better, because then you make the whole platform available to others. We're actually now, as I say, closing Stampede 1 and launching Stampede 2, partly because Stampede 1 has just become a real big beast. And one of the reasons, not the only reason, is that when the inspectors come to invest, you've got 17 years you're going back to. And of course, what you did in 2007 is to the regulations of that day, but they judge you to the regulations of today because they can't recreate the regulations of 2007. So, yeah, the whole thing is, you know, it's just, and, you know, to have those sorts of conversations just drives you bonkers. And so, so there is a time that you want to move on, and hopefully this is the time. And the oversight committees are key. I won't spend too much time on that. One of the small things that's helpful for people doing these things is try and present this as a phase 3 trial with the opportunity to stop for lack of benefit, not a seamless phase 2-3 trial. Now you can present it as a seamless phase 2-3 trial, but then you have to do quite complicated things, and I'll talk about that in a moment. If you do design it as a phase 3 trial, it's important to stop arms for lack of activity or benefit. And whenever possible, use an intermediate outcome measure, which is more information content rather than the primary outcome measure to stop. And it doesn't need to be a surrogate. I'm happy to pick up in questions why that's the case. It doesn't need to be a surrogate in the usual sense of the word. And the reason why you might want to design it as a phase 3 trial with opportunities to stop for lack of activity or benefit rather than phase 2 is that you don't then get into the conversation when you've done the phase 2 bit of it and you go on to the next bit of it, that you're selecting an agent to go through. So you have to make some allowance for that selection in terms of either starting the clock again, which some regulators ask you to do, or downweight that data in some way because you've made the selection and overestimated the effect of the treatment. That's a statistical issue. You can get round that. There are methods, but you just don't need to have that conversation. The maths are the same if you do it as a phase 3 trial with opportunity for lack of benefit, and nobody gets excited then about the selection point. So you just don't need to go there. And a thing here we've also learned is don't do small adaptations, do big ones. Don't. And adaptive randomization is, well, at least my opinion, not very helpful here. That really doesn't help. That's just tinkering around the edges. Do big things, add arms or drop arms, or amend the eligibility criteria in a big way. Don't do small things. It really isn't worth it. You're asking lots of people to do a huge amount of work for really small benefits. And you can actually then evolve this trial, which is what Stampede has evolved into, targeting subgroups of patients prospective and retrospective. How am I doing for time?
7 minutes.
Okay, that's fine. And that takes you into an umbrella trial. One of the things that we've been very clear about, and you'll have seen it when we showed you all the interventions, is to choose research interventions where it's different as possible from each other. Number of reasons for doing that. You maximize, it just makes sense. You maximize your chances. You're going to use the same intervention in each arm. They're going to give you pretty much the same result. Why would you do that? And as importantly, to give you a possibility that any successful interventions could be combined. They're not competing against each other. So you can make faster progress. And there's a statistical reason why you want to do this as well, which Babak will pick up on to allow you to use a pairwise rather than family-wise error rate. It also helps you plan for success, because you can then think about how you might change the control arm, as we've done in Stampede 4 times over that 17-year period. Just think it through. We're used to actually planning for failure, because we know, there are few in there who've been around a long time. And, a lot of trials don't produce positive results showing the new is better than standard. Loads of them do that. We're used to that. But plan for success. How are you going to change that control arm if those treatments are successful? For the statisticians in the audience, there's a sweet spot to start for the first lack of benefit analysis. One sided significance level of about 0.35. Just as a rule of, it doesn't have to be there, just that's a good place to start. And as I said, plan some success. And again, for the statisticians in the audience, this is great fun for statisticians because you have loads of them. You need loads of them because you have to make some changes as a consequence of the data emerging from the trial, but you've got to also make some changes as a consequence of not knowing the data from the trial. Either external data or things have changed. So you need to make very, very clear what changes you're making as a consequence of the data from the trial. That's the unblinded statisticians. And the blinded statisticians help you with making changes that are independent of the data from the trial. Adding arms, for example, changing the targeted effect size, changing the accrual, the event rate, because the event rate is small, changing the target to the number of patients. So you can see that actually this is very heavy in terms of staff, both all the site staff, all the central coordinator staff, but also in terms of statisticians. And you can actually do unplanned adaptations as well. You just got to plan for unplanned adaptations. So in Stampede, what we did was a number of months before the analysis, for patients with metastatic disease, put in a powered subgroup analysis, pre-powered, pre-specified subgroup analysis, before we had seen any of the results, in a blinded way, targeting patients with either low burden or high burden disease separately as subgroups. And in fact, that was a good thing to do because it's clear that only patients with low burden disease benefit. And we've also changed the primary outcome measure for patients with M0 disease. Again, you can do that if you retain the integrity of how you do it. You do it and you write it up and you date stamp it. Make sure it's date stamped in the public domain. I've said most of this. How long have I got now? 2 minutes?
Yeah. 3 minutes.
3 minutes. Okay. So now the plot on the left-hand side is just showing you a lot of the methodological papers that have come out of this. The plot on the right is showing you all the trials that have come. I've used this sort of design or some variation of it. A number of them more recently have been COVID trials, of course. When I did this slide, it was 50 randomized trials. I think there's about 75, 80 trials now using this sort of design worldwide. So it is gaining some level of use. We're applying this to a number of different areas now in neurodegenerative diseases. I won't go into this in great detail, just to say that a lot of you will know that we haven't made any progress in these diseases, MND, progressive multiple sclerosis, Parkinson's and dementia. And we set up the Accord collaboration, this collaboration of groups developing and running and reporting multi-arm, multi-stage trials in neurodegenerative diseases. And this collaboration of charities and philanthropy, a number of the partners are there, listed there. That partnership group has expanded now, and I know a number of people I can see in the audience are part of this partnership group. And there's some of the people who are helping to lead this. Where are we with this? The M&D SMART trial, which is being run out of Edinburgh. I know people from there are here today. It's now, as I said, 400 patients from 17 centers. I think that's over 550 now. The first stage analysis has been done. Both arms continued, both repurposed arms continued. Second stage analysis is planned for August, and a new research arm is being planned for September 2023. The Octopus trial in progressive multiple sclerosis has started with 2 repurposed arms, and Parkinson's disease trial has just gone in for funding, and the dementia trial has just gone in for funding, both in outline form. And what we've done there is had a more structured and systematic approach to new interventions. I won't go into this given the time. One of the things that's been really important in this, though, is again the patient input. They've been very, very clear the need for broad and inclusive eligibility criteria. And one of the things that we've added as a consequence of some of these discussions is the opportunity to re-randomize patients should an arm stop. And again, I think Susan Scott will talk a little bit more about that in her presentation. This has been a collaboration with funders as much as a funding initiative. Our funders are intimately involved in discussions about how the trials are going and how they're working and what we might do for the future. So it's quite a different sort of collaboration than the ones we're used to, I think. I'll just finish with the conclusions. We need a step change in efficiency and design, not just tinkering around the edges. I'm grateful for Camille Carroll, who had a discussion with us. It's like a 2-arm trial is like, this analogy isn't great. We couldn't get a better one. It's like building a football stadium and then only having 1 game in it and say, that's it, we're done. Not quite a great analogy. Multiple treatments being tested at the same time, things being added and dropped are challenging to design, fund and run, but they're more than worth it. Patient involvement is key to their development success. It's really heartening to see that funders now are increasingly recognizing their importance. And a patient partner has asked me, why don't you do all your trials in this way? And of course, welcome in discussion if somebody has a good answer to that question. I'd just like to thank everybody who I've worked with in this area.
Thank you, Matt.